on artificial light at night and insect behavior in Haliburton County. Just big thanks to PLCA, kind of our, I guess, contact there, Harvey Bischoff, who kind of led us on his property and showed us around the lake. And big thanks as well to Supervisor David, and also Frank and Jack for kind of their help on the project. So we could probably move into the slides here.
So we chose to do our community-based research this year on artificial light at night and insect behavior in Haliburton County. And as mentioned, we did it with Paudash Lake Conservation Association, specifically at 3 sites on Paudash Lake, which we're very kindly allowed to be on people's private cottage properties for. And we worked with Frank and Jack from Wyonch, and our supervisor was David Beresford from Trent. He was very helpful for this project with us. So the main problem we needed to identify is it was identified in Paudash Lake's management plan from 2004. Paudash Lake's located just south of Bancroft and is on the Canadian Shield. And our partner, Paudash Lake Conservation Association, was established in 1973. They identified light pollution as a concern, and they identified both ecological and atmospheric light pollution, which is those 2 things affecting insects and other wildlife, as well as atmospheric light pollution, which is that sky glow issue a lot of people experience when they can't see the stars at night. So we wanted to look at those 2 main things, how people perceive artificial light at night and also what impacts it had on the wildlife. Specifically, we focused on insects. So our main question was, how does the color of artificial light at night from cottages and other sources affect insect attraction around Paudash Lake? And we had hypothesized that cooler colored white artificial light at night will attract significantly more insects than warmer colored. And we also asked, how do cottagers on Paudash Lake perceive artificial light pollution? And we wanted to know what their awareness of potential environmental impacts and associated with artificial light pollution was. And we had hypothesized that cottagers will perceive it as an environmental problem, and most will be aware of potential impacts, but we weren't too sure what we would expect from most people on the lake. So as I mentioned, Paudash Lake did a lake management plan survey in 2004, and from about 31% of those surveyed, they indicated that they were either significantly or moderately impacted by light pollution on Paudash Lake. And the main issue here again was they highlighted the reduced visibility of the stars and kind of natural ambiance on the lake that you experience when there's lots of light pollution. So for our study, we wanted to assess how this attraction is impacted by different wavelengths of light. So we conducted field sampling in August and September of last year. And we assessed 4 different colors of white light. And you can see in the graph down there, it kind of shows roughly what those colors look like, ranging from warmer to a neutral kind of light color and cooler light colors. So we assessed 3 different sites, and we used sweep netting methods to catch insects as they were buzzing around our light traps, also using count data. So we performed stats in paleontological statistics, a statistical program that Dave showed us, and using Excel to generate our bar graphs and everything. And then also the survey was distributed by Paudash Lake Conservation Association to its members through Facebook and email. We received 72 total responses, and the data was shared with us for analysis and to see kind of what people thought about light pollution on the lake.
Right, yeah, and kind of just getting into now what actually we found from this study. And the first graph you can see here is a bar graph really of the mean number of insects that were captured per light treatment that we used. And you can see the warmer light treatment is on the left, and then the cooler is on the right. And so there's kind of a visible trend here where the cooler light treatments did attract more insects on average. But we kind of performed the stats, and through kind of an ANOVA, we found that actually there is significantly less attraction for the warmest wavelength, so the 2700 Kelvin light temperature there compared to all of the other ones. And then kind of addressing our second hypothesis, and from the data of the survey, we found here that about 70% of people agreed to the statement that artificial light at night had negative impacts on the environment, which was kind of an interesting finding here, that, I don't know, the majority of people do see that there are negative impacts to artificial lighting on the environment around Paudash Lake. And then kind of continuing on to the survey, linking it a little bit to the past thing, this question looked at more what awareness people have about the impacts of using artificial light at night. And The majority of people, again, were either aware or somewhat aware. So on a scale of 1 to 10, from 7 to 10, like approximately 2/3 of people were aware that there was impacts of using artificial light at night, but there were still just over a third of people who were unaware based on that scale. And that kind of indicates to us, or indicated to us, that there might be some sort of form of education needed to kind of highlight the role of impacts of artificial light at night and what impacts could be have. And that was echoed, I think, in a few statements from PLCA members. So they were able to voice their concerns and voice kind of some directions forward about artificial light at night. And so this person here kind of identified that education is needed, especially for modern cottagers and younger cottagers who have extensive lighting systems and maybe don't understand really the impact with that. And then something else that's interesting that we found from these concerns is that a lot of residents have major concerns about, one, not being able to see the stars and not being able to look up the night sky and kind of do that observation of the night sky, and two, that artificial light pollution might have some pretty significant effects on birds, owls, loons, and other nocturnal animals. So that was interesting. So really, what does this all mean? This means that artificial light at night, specifically ecological light pollution, might be affecting nocturnal insect behavior around Potash Lake, because they're all kind of getting attracted to lights that are set up outside at night. And interestingly, warmer colored white light would have a lower impact compared to cooler white light on insect attraction, and then fewer ecosystem impacts. And really, this is important because it has trickle-down effects on the food web. So anecdotally, kind of through our survey, we found, or through our study rather, we found that spiders would often start to kind of build nests and stuff around the lights, and we saw toads a couple times. And I don't know, really the attraction of insects to light has impacts on what food is available for the rest of the ecosystem and kind of around Potash Lake and other lake systems. So your fish should be affected by a lack of maybe food resources. And another quote from a member here, seeing that since the 60s, there has kind of been a decline or an observed decline in the amount of insects around the lake. And also answering our second hypothesis that members are generally aware of and agree that artificial light at night has negative impacts on the environment. While education might be useful, there is potentially a willingness to change light use behavior, which would be important moving forward. Kind of some of the key limitations to the study, though, were that one of our light bulbs, based on availability of local hardware stores, was a CFL bulb compared to LED for the other three, and that was CFL one was the coolest wavelength, and so CFL typically attract more insects, so that might have a confounding factor, and then sampling seasons as well. So most of you guys probably know that key bug season in Haliburton and in Ontario is like late May, early June, and kind of even going into July, but we didn't really catch those seasons in our sampling events. And so we'd expect maybe there's just more attraction at that, but that's something that needs to be looked at.
So through our study and everything that we found, we developed a infographic to send out to the members of PLCA and the wider Potash Lake and Haliburton community. Just kind of going over brief information about what is light pollution and what impacts it could have on the lake ecosystem, specifically highlighting those food web implications on specifically like bird and fish populations, as insects are key food resources for them. And we just included our bar chart that we developed and kind of the two main benefits that people seem to care most about in the survey, which was improved night sky visibility. So we highlighted that using directional shades and warmer light types can help preserve that kind of lake natural ambiance that people are looking for. And also, I'm sure we're all in agreement that having fewer insects buzzing around your cottage, and specifically mosquitoes, is a bit nicer and can improve the enjoyment of your cottage for lots of people. So our main recommendations were to switch from cool white lights to warm white. And then also from our literature review, we identified that using more directional lighting can help impact that sky glow issue that is experienced. This is just like having shades and things that are pointing the lights downward and focusing them on where you actually need lighting rather than having light scatter up into the sky. So that was kind of our key thing to hopefully educate a few people on just what light pollution is and what they can have a small change to make it a little bit better for the lake as a whole.
Yeah, and then kind of Elle and I really enjoyed this research and kind of had a great time going out and looking at the bugs. And it's relevant, one, for the environmental kind of reasons, and two, for, I don't know, there are multiple economic and social aspects as well of lighting that can be studied. And we found, and looking in the literature, that artificial light at night and light pollution is actually a chronically understudied issue. And so if there's anybody who's interested in kind of light pollution and potentially the effects that are being had on lakes and in the region of Haliburton County, then this could be a direction for future community-based research.
Thanks, Frank. So yeah, I wanted to thank Jeff Clark, who was the Trent University supervisor for the project. Also wanted to reach out to Roxanne Casey and Simon Payne from Canoe FM. Wonderful working with them both as well as yourself there, Frank. I know that we had a lot of overlap with that as well with this project. I focused this project on, it was a data analysis project with a focus on identifying listener patterns, standout shows, anomalies within the listener data that we had provided to us. We had two sources of data that we were using for this project. So the first one was a Canoe FM survey that had been conducted, and in around October, November, they had about 202 people respond back to their 13-question survey. Now, not everybody answered every single question, so the numbers of answers varied on each question from about 100 to 200 kind of things. The other one that they had invested in was Stats Radio, and this organization collects listener data for kind of every single hour within the day. The data set that I was provided by Canoe FM had it from the start of their contract, which was January 1st in 2023, up to the point when we were working on this project, which was about May 31st, 2024. All right, so lots of stuff happens with applied modeling, quantitative analysis. I was doing analysis on these two data sets, involved loading the data, preparing, summarizing that. There was a lot of questions that had text entry boxes, but kind of asked like, well, what is your first favorite show? And then also list your other favorite shows. So kind of manipulating that data to be a little bit easier to conduct analysis on was part of the preparatory steps that I was taking. Really what we wanted to focus on in this project was we wanted to kind of have the results help guide Canoe FM's some of their programming decisions, sales strategies, and potentially marketing efforts. All right, so a couple interesting things came from this project. So this first visual here really kind of speaks to the listener survey that was conducted and which shows related to which other shows. So survey respondents were asked to rate their first favorite show and rate their second favorite show. This image shows the highlights of what people identified. So the darker the blue square means that more people identified those two programs as their favorite first and second one. So some of these were related pairs, such as Canoeing in the Country and Country Crossroads. Those are two radio programs that Canoe FM has, and those ones are very related. So that one had a fairly high level of relationship between it. Another one was Only Rock and Roll and Rocking the Canoe. So something that kind of came out of this was the possibility of pairing and promoting these related shows that would help with their marketing strategies, that they could kind of tailor their advertisements during those two shows to relate to that type of listener. Another one was if they ended up pairing those types of shows back to back in their radio programming schedule, they might retain listeners at a greater or higher level than they would if those shows were spread apart, say on different days. This one showed some of the anomalies that was going on. This is a very busy infographic. It shows each hour of the day compared to every other hour of the day. So if you kind of slide your fingers along, what you'll see is that they'll connect at a square, and the deeper the red means that listeners tune in between those 2 hours. So that diagonal red line that you're seeing there, if you slide your fingers along, you'll see that those ones are all the same hour, that's 1 p.m. and 1 p.m. And that's why there's only ones in those squares, because if you're listening at 1 p.m., you're clearly listening at 1 p.m. So when we get away from that diagonal a little bit, it's quite interesting. You still see some dark red because if you're listening at 1 p.m., it's very likely that you would remain tuned in at 2 p.m. So that's why we're getting some red, but not quite as deep red as if you're listening within the same hour. So you see that there's a red box in the middle there, and what that kind of shows and demonstrates is that between the hours of kind of like 9 a.m. and 3 p.m., that your listeners are staying tuned in. It's kind of your work day, and people are leaving the radio station tuned in during those hours. So it's quite red around that area, and it makes sense that people would leave the radio stations on, kind of thing. What you'll also know is just above that center red box there, to the above and just to the left, there's actually some blue pools in there where the the boxes get a little bit more blue than they otherwise would. And that was something interesting that the stats radio data had revealed to us, because in those squares, what's happening is people who listen at one of those hours are actually not very likely to listen at the other one. And those two blue pools are somewhere where we wanted to do a little bit deeper analysis to understand better why people would not be tuning in between those two shows. And that's for a future project with Canoe FM.
I'd just like to thank my supervisor, Julian Ahern, as well as you, some of my close contacts, Frank and Jack, and everyone who helped to sample on their respective lakes. I'm sure they know who they are. Microplastic trends we've found across our lakes that we've sampled so far. And so to set the stage, what we currently know about microplastics today is that they're formed from the degradation of large plastic items in our environment. And we found them to be ubiquitous or practically everywhere around our globe in all of our environments, which is quite concerning given we also know that they are both easily inhaled and ingested by either us or animals. However, for questions guiding our current research, we specifically want to know where we might find a certain amount of microplastics, whether they're higher in one environment or lower in the other. And we also want to know the different types or the different characteristics of microplastics and how they may differ per environment. And so to give a bit more background on what exactly a microplastic is, they're broadly defined as plastic particles under 5 millimeters in length, but they can further be classified by their shape, such as a fiber, a fragment, or a foam, their specific size, their color, or their plastic type. Sometimes we refer to it as their polymer type. Evidence in humans as a direct response to microplastics is largely unknown, so we don't really know how microplastics might affect our health. But we do know in namely aquatic species, there's a variety of adverse effects from microplastic contamination. However, these can ultimately depend on the concentration of microplastics in the environment as well as their composition. Therefore, it's quite practical that we assess this in the environments we may encounter them in. In the case of our rural lakes, where might plastics come from? Well, we know that fishing and water recreation directly on the lake can generate microplastics into the lake, but also in terms of rural lakes, they can receive microplastics from a large array of regional activities, as microplastics can easily travel through either waterways or the atmosphere and be deposited into these lakes. And when a microplastic comes to a rural lake, it firstly can exist in the surface water, and depending on its density, it can either remain in the surface water or sink and be buried in the sediment. However, we'll be focusing on surface water because it's in this area that they're more at risk for being either inhaled or ingested by us or animals. So as for specifically our Haliburton lakes, we do know from past research that microplastics are present here. But what we want to know in this case is whether their abundance or their composition or their microplastic characteristics have been changing over time, whether that be increasing or decreasing, or these lakes have been favoring a certain type of microplastic over the other. So therefore, the primary objective of my study was to determine any seasonal changes in microplastic abundance and characteristics, in the Haliburton County lakes. So far we have 18 lakes that we've been sampling, and these samples have been taken during the Woodlands and Waterways Eco-Watch monitoring program from ULinks. And we've taken these samples for this presentation. I'll be focusing on 2022 to 2023 samples. And within these 2 years, we've taken them seasonally in the spring season and the autumn season. So what we do with these samples first in the lab is we separate any large organic matter or sediment from them by passing them through a 20 micron sieve. And we break this sediment down using hydrogen peroxide, and then we filter both the broken down material as well as the water itself using vacuum filtration just onto filter papers. And then the next step is to examine these filter papers under a stereo microscope, and we judge particles based on characteristics common to plastics. So that relates to their color and their texture, their malleability. And then we also test their melting point with a very small soldering iron. And we find that plastics are much easier to melt rather than the organic material or sediment we might find. And then the last step of the process in the picture on the right there, we use spectroscopy to determine the plastic type or the polymer type. And spectroscopy basically measures the amount of light a plastic particle may absorb or reflect. And this measurement can match to a variety of different polymer types the plastic may be. So yeah, I'm very excited to share some preliminary data on the 2022 to 2023 years. In terms of overall microplastic concentration across all the lakes and across the 2 years, we found that there was an average of about 3 microplastics per liter. And we found quite a large range from the whiskers on the plot there. The range went from less than 1 particle per liter all the way to almost 8 particles per liter. However, in terms of any significant change across the seasons and the years, we could not detect any. And for microplastic size, we found a median size of about 0.64 millimeters, and we found a quite large range from less than 0.2 millimeters all the way to almost 1.7 millimeters. However, in terms of microplastics, considering they can go up to 5 millimeters, the range is actually on the smaller end of particles in these lakes. But like concentration, in terms of any seasonal changes, there was no significant difference we could detect. And then in terms of the microplastic shapes we found, most of the lakes were dominated by fibers, followed by fragments, and then there was a small proportion of other shapes, primarily beads or films. However, this trend of fibers, fragments, and then other that continued throughout the 2 years, and like our other variables, we did not find any significant difference. And lastly, I was able to take a small subsample recently and determine polymer type. So in no connection to any specific lakes, this is kind of an overall look at the particles we found. We found that polyethylene terephthalate and polypropylene dominated the subsample, taking up over 75% of it. And as for what these plastics are, polyethylene terephthalate is commonly sourced from either clothing or bottles and other containers. And then polypropylene can be from containers as well, but is also found in piping and other construction materials, and maybe concerningly, ropes as well. So we can compare our results to our broad region, as there has been past research in Muskoka Haliburton lakes as well as Lake Scugog. And these studies found polyethylene terephthalate dominant polymers as well as fiber dominance as the shape. However, they only found 1 to 2 microplastics per liter compared to our 3 microplastics per liter. So what this might imply is that this study that we're doing might be a more accurate representation, as this past research was older, and since then we've refined our methods quite a bit. So in terms of what we can do, it's a bit too early from our data to first tell if there is any significant changes happening in these lakes right now. And it's a bit too early to determine any particularly major source of microplastics into any of the lakes. So what we can do in the meantime is limit ourselves as sources. And first, this could just be simply learning to kind of understand your plastic use better, understand the products you use and whether plastic may be in them. For example, some people may not know that the Walmart bag shown on screen there, while reusable, is made partly of plastic, and it can be a major source of microplastics. And then furthermore, we can continue to care about our environment. We can encourage cleanup initiatives such as our shoreline cleanups and get rid of that improperly disposed plastic around our lakes that may generate microplastics. And then just as importantly, as we're doing now, we can stay up to date with the research, and we can communicate our findings to our community, to our peers, because awareness strengthens further research and eventually solutions to our problems. And so this project is far from done. We're going to continue sampling seasonally until 2026. I'm currently analyzing the 2024 samples, and we're working on collecting 2025. And my hope is in the end, we'll be able to create a sort of regional and long-term monitor of microplastic contamination that would spatially be among the first of its kind in microplastic research. And then also excitingly, kind of support new projects and expand this analysis of microplastic kind of monitoring and contamination to all 25 lake associations. As we have the samples, it's just a matter of increasing that bandwidth to be able to analyze them.
So today I'm going to talk about some aspects of time and timing on approaching the problems related to invasive plants and how we can better management. But before I start, I'd like to acknowledge that I work on the campuses of Algoma University, their traditional lands of several First Nations, and I work in Robinson Huron Treaty territory, home of Garden River and Batchewana First Nations, on lands that were set aside for education as envisioned by Chief Shingwak here in the picture. This was nearly 200 years ago. He was so well ahead of his time, and he had a clear understanding of the true value of cross-cultural teaching and learning, and did the best he could to try to set land aside and to ask for schools to be built for his people. So invasive plants for an invasive, and I teach two courses at Algoma: invasive species biology and climate change biology is two of them. And I start with this always with this hand here when I teach invasive species biology. For a species to become invasive, there's good reasons why non-native species have a higher probability of becoming invasive. They need to arrive internationally. They need to reproduce alone, go through all these filters, right? So they need to spread away from their parents, and then they need to reach populations that have high density. And then there has to be some kind of negative impact. This could be environmental impacts, this could be economic impacts, and social impacts as well. I think all of these need to be considered as a whole in a broader capacity. Now, we can't isolate things like invasive species and the problem of invaders. So I'd like to start with global change drivers and starting with climate change. This slide just basically introduces the global change factors. Climate change here, we know that the average temperature is 1.1 degrees higher than pre-industrial levels. We know this is unprecedented since humans have been around as a civilization, even as a species. It could go back in terms of CO2 levels all the way up to 800,000 years if we look at ice cores. And we know from the Paris Agreement, there's good reasons why 1.5 degrees on average is problematic. And we can run into runaway climate climate change potentially once the permafrost starts going and all that carbon starts going into the atmosphere. We know that warming is uneven, and that in northern latitudes, warming is more is going on for the climate. The planet is warming faster in northern latitudes, and you could see that here. How the planet is warming, and you will see it over time, decade after decade. You can see that it's warming much faster even in Canada here, and of course, just a little bit northern from us. You'll see all these red spots much higher than the 1.1 eventually over there. So we need to consider this. What's happening here as well is that plant communities are changing. The ranges of plant species, of native plant species are changing. And this is an example here. You could see for aspen, the current climate suitability zone, and where between 71 and 2000, and where we expect for this particular species, where we expect it will be in 2000, between 2071 and 100. And obviously, with the shifting of plants and the suitability areas for plants, there are empty niches that are being left behind. And what kinds of species will take on these niches is we don't know very well. We don't know if species that are in southern latitudes can migrate north if they're through assisted migration, and will they be able to establish or not, or will we see an increase in invasive plants establishing? So these are all unprecedented circumstances that we will have to deal with. Aside from climate change in terms of global factors, and glad we just heard about microplastics, there's pesticides, heavy metal, plastics, and also aspects like nitrogen deposition, right? That occurs when fossil fuel combustion and intensive agriculture release nitrogen that then falls into the ground. We know that invasive nitrophilic plants, for example, they have a competitive edge in nutrient-poor habitats. And there is recent research raising hypotheses about how invasive plants may gain an advantage relative to non-native plants when it comes to competitive interactions in the context of environmental pollution. So we really need to consider all these global change factors together with invasions. Now, invasions are here, and some of us might think that with increasing trade, or that we have somehow reached some kind of plateau in trade, and we have potentially maximized, or the number of species coming in has plateaued somehow, but we know this is not true. And you could see this here in this data set from PISEC and colleagues. And so you see for vascular plants, mammals, fishes, all sorts of different types of organisms across the different continents, the numbers are cumulative numbers of species richness, invasive species richness is nowhere near saturation. And we should expect to continue to see novel introductions and establishment. So we know that invasive species is one of the top threats of biodiversity, but worldwide, there's all this habitat fragmentation, then climate change, then you get all these invasive species. And so managing is, in the face of other global pressures, a very pressing issue. Now, transitioning here, what's the Ontario invasive plant situation? So Ontario is a hotspot for invasive species in Canada for invasive plants. Ontario actually has the unfortunate distinction of hosting the highest number of invasive plants in Canada. Aquatic invasives are also a concern. But so why so many species in Ontario? A combination of factors. Canada is the most populous Ontario is the most populous and industrialized region, and 64% actually of overseas shipping containers entering Canada, they come through the Great Lakes and the St. Lawrence corridor. If we look more deeply at vectors and pathways, and you could see that in this paper here by Trublin et al. In 2006. And there's these network graphs are done for intentional and unintentional introductions, and looking at North America here for intentional introductions. If you try to see where or what pathways, what kinds of are involved, you see that for intentional introductions, most of the invasive plants being introduced and establishing come from the ag, horticultural sector, ornamental plants. And then when it comes to unintentional introductions, most of it, yeah, there's a little bit from shipping containers, ballast water, but the majority falls into what's called ignorant possessions here. So these are seeds that come with other plant products, garden supplies, and things of that sort. So that's mostly from the literature. We have a very good grasp as to where these plants are coming from and where we need to keep vigilant. When I started my position here at Algoma, there wasn't really a guidebook for the region, and that was one of the first things I started working on with the student Lisa Derricks. And so this book is available. You have the QR code here. It's free of charge. It's a PDF you can just download. And in this book, we selected 14 invasive plants we were mostly concerned with. This is about 10 years ago, a little bit over now. There were definitely some other species that I potentially could consider, but these are still the most important. So we had 14 invasive plants, you see them here, and also we included 11 invasive insects and pathogens that we consider to be of the highest priority in Ontario. For all of these here, the reason, how did we get to prioritize this particular species? We used an approach looking at economic risks, environmental risks, social risks, and for each one of these risks, we broke them down, as you see here, and we looked for evidence in the literature, actual data that would allow us to prioritize, create check marks and create priorities. So we did this for a huge variety of species, and then decided to include these ones hit the most check marks. And so for each species, and you see this for Barbary and English ivy, you could actually go through the specific risk and then get to the root of the literature and look at the evidence and what papers are involved. Now I want to go from transitioning from this background area of my talk into looking at the timing of invasions and look at the invasive species curve. And I'm going to focus today in two areas of the invasive species curve. The invasive species curve, basically on the x-axis, you have time since a species is introduced. Then at first, we don't know the species is there. By the time we usually detect it, there's still a chance we may be able to eradicate this species, but a lot of times it could be too late, and then only some kind of management is possible. And so today I'm going to look at two areas of the invasive species curve. This early phase where there's a lag time, and some species could be non-native, naturalized even for many decades before they start to become invasive. And then other species may have much shorter lifetimes, and species that have short lifetimes, those are probably the ones we should be prioritizing. So how do we know what species have a long lifetime versus a very fast invasion capacity? That's one area I'd like to discuss today. And then also what happens once they reach high abundance and after quite a while. So will they persist indefinitely at high as invasives, or will pathogens start catching on, diseases emerging, and we will start seeing population level declines? So these are things that we want to know for different species so we could potentially prioritize management. And that's I want to dwell a little bit on this today. So one of the first things I did here with a colleague here was to we started thinking about this early, the lag times, and we wanted to develop standardized and comparable invasion curves for extant native and non-native species using herbarium data. And as you probably might know, that herbaria, there's less and less funding for herbaria, and these are very important resources. And having herbaria and having them well stocked and projects ongoing, monitoring plants and in a certain region every year in a strategic way is I think very important. And getting funding for herbaria in this context is, as I will try to show why we think this is relevant at a time when funding is declining for herbaria. So as I mentioned, with the invasive species curve, you can see here there could be two hypothetical situations, two different species, one that has a lag time, let's say, that is the yellow line here has a much longer lag time, and one that starts spiking much faster here in the dashed line. So how do we distinguish the two? How do we catch this ahead of time before we know what kind of trajectory these the species we have in our region, say, are going to start spiking and becoming potentially invasive? So a methodology that could be used in herbaria is, of course, continue the data collection, adds lots of supplementary resources, metadata, identify the key stages of invasion for each species. So actually starting putting numbers on that invasive species curve on the time, and then in terms of the y-axis as well of population density. And then analysis of biases and standardization challenges. We in this paper, we explain how this can be done, and then integrate spatial and temporal patterns as well. What kinds of quadrats, what kinds of spatial area do you go out every year and sample the species into your herbarium? And then what do you do with this data? And one of the things that could be done is, for example, these proportion curves, where you have, say, a native species as a baseline here, and then you also have non-native species. Of course, a non-native species starts, that the orange dash line starts much lower because at first it hasn't been introduced. And then as these invasive species start to establish and their populations start growing, expanding, becoming denser, it will at some point surpass the line of the native species. And so you could have an overall line, if you can see that all the data set of your herbarium records, and which is that red proportion curve line. And based on those lines, on these data, you could start making inferences and saying, hey, this line is becoming really big year after year. Let's see what species are moving this needle more than others. And then you're targeting these species for control, because resources are limited. So this is why you're gathering all of this data and creating these proportion curves. So this is all very conceptual things that have not been implemented yet. These are ideas and that still need to be implemented. So let's have how could we also build this from a variety of traits. So in here, it's also hypothetical. You have the lag times on the y-axis, and then you have several different species here. And you have known invaders, for example, that are present, but you have a species that is not yet even established, hasn't even entered Canada, is not present, but you want to potentially, it could come in either for reasons of trade, etc., and you want to know the traits. And here, the only trait I have is growth rates. And this growth rate is on the axis on the right-hand side of this graph. So you can see that if a species has a growth rate that is very low, then the lag time, of course, is going to be much higher. But this is just growth rate. There could be a huge variety of other traits that we can integrate in these kinds of data set, which more clearly will then give us an opportunity to anticipate, to predict, okay, what type of lag time do we expect? Many decades? Could it be 2 or 3 decades? This species here, we should really be on the watch out for this one in particular. There are some groups, and there's a few very well-known cases in the United States, for example, with invasive algae, where even some people, citizen scientists, are have boots on the ground. They are looking for species that are not even present there, and when they see them, when they finally comes in, they've detected them, eradicated them very quickly. So this type of framework could help in that regard. So standardized invasive curves may enhance our ability to predict and manage invasive species. Collaboration across herbaria and research networks is essential, but future work should focus on refining methods and integrating diverse data sources. So next, I'm going to talk about move to the time later on at the invasive species curve and talk a little bit about some work from my group that we've done on the vulnerability of non-native invasive plants to novel pathogen attack. Do the plant traits matter? Can we predict what kinds of invasive plants will potentially start declining due to accruing enemies? And so, of course, this is the same invasive species curve. I just added, as an example, a couple of traits there. So we could have, say, persistent, if the plants persist over time, they maybe have traits that allow them to be more resistant to enemies, say, specific leaf area. If they have very high specific leaf area, not a lot of surface for pathogens to come into the leaves, then they might persist more. And this is obviously just one trait. But if it looks more like a banana tree leaf, maybe this will get more pestered with pathogens that are capable of causing declines in fitness and the ability of these plants to produce seed and offspring. This is what we actually want to see. So can looking into traits and creating some kind of framework help us eventually prioritize? Now, over the last decade, there's increasing evidence suggesting that pathogens have the potential to regulate long-term dynamics of invasive plants through the emergence of novel interactions with pathogens that are capable of reducing the invader's fitness. So basically, pathogen accumulation. And there is some evidence that the longer a population is present at a site, and this data set here is a data set from New Zealand, as the smaller the growth rate it has, and this could mediate actually plant pathogen interactions. So here what you see is that, and it's a study from, we've known this for over 10 years, that negative feedback, so a plant growing in its own soil year after year after year, it can correlate with the years since the invasive plant establishment. So the longer a plant is established, the more enemies is building below ground in this case, and when exposed to its own soil, it grows less than if it's exposed to soil where this plant has not been present. And that's why you see these are each dot is a different species. Then you have the year since establishment, and you see that the longer that establishment, the lower that feedback. So based on this evidence, what we started doing, we used the TRI database. We looked at plant functional traits, and these are traits that impact the fitness indirectly through effects on growth, reproduction, survival. And first we created trait categories that we thought were likely to determine the resistance and tolerance of plants to pathogen attack. And they are all listed here. So next, for each trait, we looked for evidence that it can be involved in some kind of pathogen resistance or tolerance. For example, for stomata, it has been shown that lower stomatal density can prevent pathogen infection. It makes sense, right? So there's less points of entry for that pathogen to come in. Also, pathogen closure in response to pathogen presence reduces infection. So that obviously is a good trait for us to include. And so we created these lists of traits. Then next, we looked for evidence of novel pathogen invader interactions in the literature, knowing that there was no synthesis of documented examples of novel pathogen attack of invasive plant species. So to determine the prevalence of these novel interactions, basically we compiled data on plant invader disease from published studies. We searched the literature, we found 661 studies, and then we painstakingly went through all of these papers with a few conditions. For example, plant species must be considered non-native invasives. Putative pathogens must have been found in a non-native range of the invader. Another condition was that the effect of the putative pathogen must have been tested by inoculating the invader with the pathogen, and then the pathogens must be native to the non-native range of the plant invader or globally distributed. So again, we compiled all of these data, and there's not a lot. This is a start. We identified 31 invasive plant pathogen combinations with 31 different invasive plant species that were experiencing novel pathogen effects from actually 27 unique pathogens. And this, we found 14 forbs and then some vines, 6 vines and 4 leguminous shrubs. Here you could see, for example, for our famous or infamous garlic mustard, we also found some records, and so with these data, we could actually take a look at what kind of pathogen and how much these pathogens have the potential to cause reductions in fitness. Then we were able to aggregate all of these data, and we collected on pathogen impacts. And here, from the previous table, we applied this multivariate model to associate the plant traits with the pathogen effects. So here you see all the different plant species that had novel pathogen interactions, and you see the most important traits. So basically, again, we compiled data from the TRI database for each invader, and then we selected all the traits, and they included, you see them here, plant height, leaf nitrogen content, seed mass, specific leaf area, and leaf dry matter content. So we calculated the pathogen effect as the relative difference in fitness between the control and then the pathogen infected plants averaged across all available studies of each plant pathogen combination. So even though we have very limited data, from the very limited data, we were able to collect these traits and demonstrate that they can actually be useful to some extent in determining pathogen effects when included as part of a much broader network. So many of the traits identified as important, they were not available for the invaders we identified, but still we found evidence for these 31 cases here, and these traits that are here were all significant. So to summarize, from this framework, we can actually begin to determine the trait axes that are most associated with pathogen vulnerability. So we found that together, plant height, nitrogen content, seed mass, specific leaf area, and leaf dry matter can be useful in trying to pinpoint what kinds of invasive plants may eventually become more susceptible to pathogen attack and then start declining without us having to intervene. However, there's a caveat here. Of course, we can't just be waiting for nature. And it might take, for some species, we have evidence it could take too many decades, and there's already too much harm caused by then. So I wanna make sure that I understand that, and this is not a magic bullet or anything like that. More trait data for invasive plants are needed to keep refining. That's exactly what we need. Keep refining the predictive capacity of the proposed framework, and then understanding the invasive plant pathogen interactions can lead to breakthroughs, both at fundamental and management decision-making levels. So with this, we just need better monitoring. We always report invasive plants, but very much more rarely we report the diseases they may have. If the leaves are damaged, if there are insects chewing on these leaves, which ones are they? Where are these populations potentially located? There's a lot less data on all of that. To conclude my talk, in terms of integration for the future, Short-term wins are possible. The good news is that with vigilance and timely action, we can manage and even eradicate certain invasive species at local scales. Success exists. But long-term pressures are mounting, as I mentioned in the beginning: climate change, the environmental pollution, habitat disturbance. All of these continue to intensify. And no amount of invasive species management alone can fully overcome the climates that's becoming more inhospitable for invaders and stressful to invaders. So we need better integrated solutions. We have to address the root causes in parallel with managing symptoms. So mitigating climate change, reducing greenhouse gas emissions, curbing pollution and nutrient runoff, restoring habitats and build resilience. And how can we get there? Collaboration and policy. It all hinges on that. Scientists, land managers, policymakers, the public, things like this. What we're doing here today is exactly the kinds of things that are part of the solution. Policies should also be aligned, policies that are tied to bringing ornamental species or agricultural crops. This all needs to be aligned. The left hand has to know what the right hand is doing. Public engagement is crucial, of course, and time does matter. I mean, this is the whole point of the talk about timing, and when do we need to act here? We know that overall we need to act. There's urgency here for integration, but there are things we can do. And maybe AI can help us to some extent to develop some of these models further so we can establish priorities. So with that, I want to acknowledge my funders, the Canada Research Chairs Program and CERC for the grants, and everyone in the Plant and Soil Ecology Lab. So thank you very much.
I chose today to speak not so much about my own research, but rather about the role that community members have in participating in aquatic science. I'm going to try to keep it really succinct. I love to gab about aquatic plants, but we'll try to keep it to the point today. So as Andy mentioned, my work usually involves working with community members, lake associations, different organizations, as well as research through the university. And generally what I focus on is aquatic plant community composition. And much of this work comes from an interest in the presence of invasive species in a lake, and kind of an interest in or concern of the alteration to dynamics in the community that they're being introduced to. And so I'm going to share with you a story about one of the surveys that I did a few years back in Gilchrist Bay. My research partner Lucy and I were there kind of mapping the distribution and relative density of starry stonewort, which I'm sure most of you know, but is an invasive aquatic macroalgae. And on one of the days we were out surveying, we kind of noticed a woman on the sidelines, or on the shorelines rather, waving us over. And so we went over and she asked us what we were doing. So we shared with her kind of our project and how we're interested in starry stonewort. And so she said, come on over. I've actually been pulling out starry stonewort most of the afternoon. And so we went over, and on her dock was this pile of green, but it wasn't actually starry stonewort. It was plants that looked like this. And so these are all native species that could be typically found in a back bay on Stony Lake or anywhere kind of in the Haliburton area. And they were doing a really good job of blanketing the bottom of, or blanketing the sediment in her back bay. And so by her pulling them out, she was kind of removing the service where they might have been buffering the area to the establishment of starry stonewort. And I'm telling you this story not to place blame on this woman or to cast judgment in any way. What I think it really highlights is the importance of knowing the native plants as well as the invasive plants in your lake community. And beyond that, kind of understanding their role or their functionality within that lake ecosystem. Kind of switching gears a little bit, I'm sure this is a familiar sight to most of you. I personally don't have a cottage, but if I did, I'd be having my morning coffee there every single day that I possibly could. And this situation also presents a really unique opportunity. I want to share with you another story, but this one was presented to me in my Foundations in Indigenous Environmental Science course this year. It's a story, and it's not a big story, of Gwiwazins, who makes a lovely discovery. And this story was told to me by my teaching staff, but it can be found in Leanne Simpson's book, The Gift is in the Making. And I highly suggest that you guys check it out, because I'm just going to paraphrase it, but it has some really lovely messages in it. So Gwiwazins, which is the Anishinaabe word for boy, was out one day in the forest collecting firewood when he decided to sit down and take a bit of a rest at the base of a maple tree. And it was a really lovely spring day, and Gwiwazins was very happy, very content, and just looking around and checking out all the spring activity that was happening, and notices Sangu, or black squirrel, that was nibbling away at the bark and kind of sucking at it a little bit. And so curious, Gwiwazins does the same. And what he found was that the water that was running through the bark was lovely and it was sweet. And so he goes back to his mother and shows her the sweet water. And together with his mother and the community, they use that water to make maple syrup. And again, that's a paraphrase, so please go check out Leanne Simpson's book. But the message that I take away from that is the power of mindful observation. And in the context of lakes and invasive species in lakes, I think a lot of times we focus on invasive species, but there's more to the story than just that species itself, but the whole community is of importance. And when you have your morning coffee out on the dock there, it's a great opportunity to just look into the water and see what's happening. Notice the little details of the plant community that are there, and let that kind of generate questions for you. Why are there always little bugs in that big yellow flower and not this one? Or, why do the big fish hang out underneath the lily pads, but little fish kind of prefer milfoil stands? And, what is this new plant here? Don't recognize it. What's that? What role does it play in the ecosystem? And that new plant might be an invasive species, but what role it plays and the impact that it's having, or rather the interactions that it's having in the native plant community, is really important to observe. And for me as a researcher, those questions are really, really interesting. They kind of give context to my work, because I'm just going out and I'm visiting your lake and I'm systematically collecting these numbers that are supposed to represent ecology. But without the storytelling that happens at the dock, I don't really have a better sense of what is happening in that lake. And you folks, as property owners along the shoreline, or people who are visiting the lake consistently, have the opportunity to intimately understand and observe those relational interactions in a way that I couldn't possibly capture in my numbers. And so what I'm doing is kind of capturing one side of the story, but there's a role that could be filled by the community to capture the other side that really shows the full extent of truly what's happening within your lake ecosystems. So just a few takeaways. Mismanagement in terms of removing plants that are not the target species, for example, remove services from the lake ecosystem and can have more harm than good. Community members have a really unique opportunity to intimately get to know the lakes in a way that researchers who are visiting don't have the opportunity to do. And that information, those observations, are data. That is science, and it is really meaningful in the broader conversation. And the questions that are generated from your observations can inform research, but perhaps more practically can inform the way you go about managing a lake ecosystem. especially when it comes to the intensity at which you're managing something. Yeah. And so for you folks, for everyone, I challenge you this upcoming season to go out and to find a few plants that you don't recognize or you've seen around and don't know the name of. I challenge you to learn their names. There's plenty of resources. KLSA has a guidebook, aquatic plant guidebook. FOCA has a guidebook. iNaturalist is a really great resource as well for plant identification. But beyond that, do a little bit of digging and understand or learn the role of that plant in the general ecosystem. Getting to know that kind of information and understanding, I hope, can kind of deepen your understanding of the relationships happening within the lake itself, and perhaps strengthen the relationship that you have with those ecosystems. Thank you very much. That's all I have for you today. And thanks to everyone again for coming out.
So thank you. First of all, I'd like to make a shout out for Kaylee's is the author of the FOCA Aquatic Plant Guide that has just been released. And I also, well, I'm going to be just giving a talk that's highly focused on a couple of species. I want to echo what she just said about this notion that we really need to pay attention to the community, and that sometimes Pedro made a comment about novel ecosystems, and sometimes these plants that we think of as non-native invasive have effectively become naturalized members of our community now, offering many of the same sorts of services that some of our natives do. So this is going to be an aquatic plant focus, although there are studies from the terrestrial world that show some of those same trends. And I think, you know, one of the things, and I think when I first finished my graduate work, it sort of fell into this same perception that these, you know, the language that we use to describe these species is often quite, I think it elicits a threat response. And in fact, I was asked years ago to participate on a panel to talk about the zombie plant, and the editor from the news station contacted me and asked me a bunch of questions, and of course my response was, well, you know, it's got a lot of the same characteristics of many of our native ones, and in some lakes it's really not that bad. And I wasn't invited to participate beyond that. You know, so this is another species that Kaylee had just mentioned, starry stonewort. And we're going to come back to this at the end, but starry stonewort macroalgae that, you know, showed up, probably been around a lot longer than we realized, and we just misidentified it, but in fact kind of became quite popular and more well known. And so then there was this massive sort of threat response that went with it. The other thing that's happened over the time, Stephen Carpenter sort of put this invasion curve together when they were first talking about Eurasian milfoil appearing on the scene in the 1970s. And this is a similar curve to what Pedro just referenced, in the sense that we know that there's this sort of exponential rise in abundance, and then it's sort of some sort of a peak biomass, and then there's some sort of ecosystem regulation which happens. And our risk assessment models in the invasive species world sort of say if the ecosystem can assimilate the species quite easily, then it's a low risk species, and we kind of move on from there. And then there's other ones where the assimilation period, naturalization period takes a lot longer. And then I think a third piece that we don't really understand is
is the extent to which our efforts to try to eradicate these things, because they are invasive or non-native, actually perpetuates that exponential increase. So they're just continually trying to replace themselves, and we do a very effective job of removing adult plants, and we also remove other components in many times when we're trying to manage these things. So that community that Kaylee mentioned is often a victim of our management efforts. But the milfoil situation is one that is probably the most ubiquitous plant that everybody worries about. I know certainly in the Haliburton region, it's got everybody's attention. And it's amazing in some cases how quickly it does sort of get that sort of regulation. And these are probably pathogens, insects chewing on it. These are probably root pathogens. We know there's a couple of funguses that chew on it. So we did some lake-wide surveys. We did quite a bit of work with milfoil, originally related to herbivory stalking, the famous milfoil weevil with Big Cedar Lake, which is just north of Peterborough, north of Stony Lake, and did some whole lake surveys and had real, effective success in regulating the presence of milfoil at the local scale with weevil stalking by introducing the herbivore, but certainly didn't do much at the lake-wide scale. And part of that was lack of resilience, we thought, maybe lack of competition in the community, not a very diverse community of plants. And then lo and behold, if you go to the next slide, I was just out this past summer and there's no milfoil in the lake, or if there is, there's just a few spots. And so we see the same phenomenon with hybrid milfoil in the Kawarthas. I've heard it from other lakes. Kaylee's done some work on Twin Sisters Lake north of Marmora, where we see these whole lake collapses of milfoil. And in this case, we're not talking about long time scales, sort of a decade. And it's not just unique to milfoil. We've also done some work with fanwort, Kabumba caroliniana. In Ontario, the only lake it's found is Cashabog Lake, which is just northeast of Peterborough. And it creates these sort of dense patches. And when you evaluate the sort of the ecosystem response of those dense patches, it's different. It's similar to some of the native milfoils, but it is different than the rest of the lake. But when you evaluate a bay that has been infected with, impacted by fanwort, and that slide, the part of the slide on the right there, which just shows a bunch of crosses, so to speak, that's a food web analysis of bays that had fanwort and bays that didn't have fanwort. This is back in 2008. And no difference to the food web. So the ecosystem seems to adjust. And whereas as the, people weren't too happy with it, although it's not a bad plant to swim in. And then we revisited this lake. I don't have the data. That was on the modified version. We revisited this lake a couple of years ago in these same bays. And the bays that were historically infested with fanwort, fanwort is now just a part of the community, and the control bays that had no fanwort in it now have higher amounts of fanwort. So it's moving around, but over time, something seems to be happening to it in terms of regulating it. And these are in low developed bays, so there was no management that was going on. And so could be something due to nutrient dynamics in the sediments, but there's probably some sort of a pathogen that's a part of that story as well. And then finally, this is stealing some data from a study that was carried out in Wisconsin. And I tried to get more recent survey data, but on starry stonewort. And a lot of those articles that I discussed about starry stonewort infestation, the demonizing of starry stonewort came from local papers in Wisconsin. But what it shows, I think, if you look at the top green line, which is the only one where it's decreasing, and this is just frequency of occurrence, how on an area basis, that the only lake where there was no management happening, and that's that Pipe Lake, the triangles where it's declining abundance. Whereas all the other lakes where they were attempting to manage it continued to see increases in growth. So suggesting this notion of, do we perpetuate, do we inhibit maybe the ecosystem itself from regulating, these high amounts of starry stonewort? We've seen similar collapses in Stony Lake and in other areas of the Kawarthas. And I think, there's a podcast that was just released based from the University of Wisconsin Sea Grant, which is a very good listen, and I think something we should all pay attention to now, in the sense of terms of how we describe, how we respond to invasives. This singular focus on these singular species, sometimes we miss the bigger issues that Pedro was highlighting in his talk about some of the other factors which likely lead to increases in these abundance. But this notion that I think their ecosystems are changing, and this expectation they're always going to stay the same is not a realistic expectation. And so perhaps what we need to do is just celebrate what we do have. Anyway, and I'll leave it there. Thank you.
So this is about the mystery snail licensing program that has run since 2021 to 2025. You can see the Chinese mystery snails on the picture on the left there, and you can see them in the lake. So a little bit about the CHA. As Andy mentioned, we bring together, we're dedicated to protecting lake health. We focus on the two biggest threats to our lake that we can do something about in a concrete way, and that is septic systems and the loss of natural shorelines. We are not involved in invasive species or other issues that are tackled by other organizations, with one exception, which is this program. So Chinese mystery snails, I think many of you know about them, but they've been spreading up through the Trent system, and they're in most of our lakes that are connected to the Trent system. Females can have up to 100, can give birth to up to 100 juveniles in a given year. And normal snails, they would be an egg, so a lot of the eggs would get eaten by fish, but these snails are actually born in the shell. So it makes them very resistant to being eaten. They are a threat to lake and human health because they force out native species. They're a vector for parasites. They cause swimmer's itch and reduce recreation pleasure. On one of the lakes locally here that does not have a lake association and is not participating in this program, a contractor that was working at my house invited me to come down and take a look at the front of his property. And if he had wanted to go swimming, which he had stopped doing, he literally would have had to part the shells that were from the dead snails floating, and to step on the bottom he had to wear protective shoes because there were so many on the in the lake. So how we got involved is that a few of our lake associations had started collecting before 2021. And then they were informed by the MNR that they had to have a license to collect these, and there's reasons for that. But the license process that the MNR told them they had to go through was each person had to apply for a license. They would get a license for only collecting on one lake, and it took 2 years to get a license. So our members approached us and asked for help, and luckily we had relationships and were very well regarded and trusted by the MNR. I contacted Dave May, who was I had known for years, and we had a really good relationship, and he had a trusting relationship that what the CHA said they would do, we would do. And the Ontario Federation of Anglers and Hunters Invasive Species Awareness Program and Dave May and the legal team at the MNR, we entered into 5 months of negotiation, mainly with the legal team at the MNR, to develop a program. And then we had to work with the 4 municipalities to allow us to dispose of these once they were collected out of the lakes. So the proposed solution was a master license under a scientific collector's permit. It involved CHA and interested member associations, the MNR, and the OFAH invasive species. We started out with 235 volunteers. We had to apply for the license. Each person had to take training and be listed on the permit. They have to carry the permit with them when they're in the lake taking snails out. And as I said, we had to work with the 4 municipalities on a process of disposal that was, and euthanasia that was humane and also protected from further spread of the snails. So we started with 235 volunteers, and last year we had 425 volunteers, and we've collected over 1.3 million snails. This is a picture of one collection day on South Lake, a local lake. They had an event that morning. Not all the snails collected were in the cart at this point, but they collected over 80,000 snails that morning. There were still a number of the volunteers that did not make it to the celebration because they were still in the lake collecting. So yeah, almost 1.4 million snails removed to date.
To thank all of our sponsors, our partners. I especially want to thank all of our speakers today. The student presentations were great, and like I said, just the tip of the iceberg in terms of the knowledge that they've brought to our hosts in Haliburton County. And so again, I encourage you to go onto our website and to look at all of the other student posters. I think most of them will have email addresses possibly of the students on, but if you would like to talk to those individual students about something that's on a poster, if you can't find it on the poster, reach out to us and we'd be happy to do that. I believe the last slide is this one I mentioned at the beginning. If anybody is interested in donating to ULinks, we'll put this QR code on our website. We did lose quite a bit of money as a result of the weather situation today in terms of the lunch and the buses and the brochures that had to be printed and are not now used. If anybody does want a copy of one of these brochures, just get in touch with myself or any ULink staff, and we'll make sure you get one way or the other. So, you know, ULink staff certainly appreciates your attention. You're coming to the event. ULinks is here basically to serve the community of Haliburton. We work in all sectors: cultural, economic, social, business, and environmental. Most of our work right now is environmental in nature. But if you're an organization or even an individual in the county and you've got a problem you can't solve, come and talk to us and we'll see what we can do. So ULinks is really, really happy to be part of Haliburton County. We really appreciate you coming out today and supporting the work of the students. And again, a special thanks to the panelists who a lot of different facets and insights against a complex topic, which, as you can see, can be attacked and analyzed from many different scales. I believe we do have some small gifts of local maple syrup for our panelists, and we'll figure out some way to get one of those small gifts up to Sault Ste. Marie, Pedro, and the others distributed as well too. So if there is nothing else, again, thank you very much, and I wish you all a good day, and thank you again for your attendance.